Lipids are one of the four major biological macromolecules. Although insoluble in water, lipids are soluble in alcohols and ethers. It is the only macromolecule which is not made up of monomeric units. Lipids are divided into four different types: fats and oils, waxes, phospholipids, and steroids. Lipids are a crucial component of living organisms because they are responsible for providing energy, storage, insulation, and producing hormones the organism needs to survive. They are also a major component of cell membranes in living organisms. Our main focus today is fatty acids.
Beta oxidation, which is the process of breaking down fatty acids, and the synthesis of fatty acids are crucial metabolic processes and play many important roles in our body. When glucose is depleted, the body will start breaking down lipid molecules, specifically fatty acids, as an alternative pathway to produce energy. Meanwhile, when the carbohydrate levels are high, the fatty acid synthesis pathway is triggered to store the excess amount as fats. Beta oxidation and fatty acid synthesis seem to be the reversible reaction of one another, but they are separate pathways because each pathway uses different types of enzymes.
Beta oxidation of fatty acid is a catabolic process that involves several steps where fatty acids are broken down to produce energy. It takes place in the mitochondrial matrix of eukaryotic cells. However, when the chain of fatty acids are too long to enter the mitochondria, it can also take place in the peroxisome. In prokaryotic cells, beta oxidation takes place in the cytosol. The process is initiated through the breakdown of long-chain fatty acids, which are converted into small acyl-CoA chains. This reaction will produce acetyl-CoA, FADH, and NADH. Acetyl-CoA will enter the Krebs cycle. FADH and NADH will go through the electron chain. Following both steps, ATP will be produced and used for energy.
Beta oxidation had 4 steps. Step 1, dehydrogenation. The dehydrogenase enzyme removes 2 hydrogen from carbon at position 2 and 3, alpha and beta, forming a double bond. In this step, FAD+ is an electron acceptor to produce FADH2. The reaction produces trans delta-2 anoic-CoA. Step 2, hydration. Water molecules are used to break double bond to form L3-hydroxyacyl coenzyme. The reaction is catalyzed by anoic-CoA hydranase. Step 3, oxidation. The hydroxyl group in carbon-2 is oxidized by NAD to generate beta-ketoacyl-CoA and NADH. The reaction is catalyzed by dehydrogenase enzyme. Step 4, the beta-ketoacyl-CoA is cleaved off by the thiol group to yield acetyl-CoA and acyl-CoA. The acetyl-CoA then enter the Krebs cycle to continue the energy-making process. The enzyme required for this reaction is beta-ketothiolase. The general equation to calculate the number of beta oxidation cycles that can take place in a fatty acid chain is the number of carbon of a fatty acid divided by 2 minus 1.
Fatty acid synthesis is an anabolic reaction which occurs in the cytoplasm involving a large complex of enzymatic activities. The following steps are involved: activation, transportation of acetyl-CoA from the mitochondria to the cytosol, initiated when free acetyl-CoA accumulates within the mitochondria. The acetyl-CoA will react with oxaloacetate and the enzyme citrate synthase to form citrate, which is able to move between the mitochondria and cytosol. Once in the cytoplasm, the citrate is converted back into acetyl-CoA and oxaloacetate using ATP and the enzyme citrate lyase. Formation of malonyl-CoA. Malonyl-CoA is formed through carboxylation of acetyl-CoA using the enzyme acetyl-CoA carboxylase. This is a commitment step because malonyl-CoA serves no other metabolic role but fatty acid synthesis.
Elongation. Step 1. Acetyl-CoA and malonyl-CoA will each react in a substitution reaction to replace the CoA with the cysteine or penthanate group on the acyl carrier protein, forming acetyl-ACP and malonyl-ACP complexes respectively. No energy is required in this step. Step 2. In this joining reaction, the acetyl is transferred and joined to the malonyl-ACP group. Condensation takes place with a loss of carbon dioxide, forming acetoacetyl ACP. Step 3. hydrogen is added to the carbonyl group on carbon 3 of acetoacetyl ACP, forming beta-hydroxybutyryl ACP. Step 4. A double bond is formed between carbon 2 and 3, resulting in a loss of water and the conversion of beta-hydroxybutyryl ACP to crotonyl ACP. Step 5. Crotonyl ACP is reduced through the hydrogenation of carbons 2 and 3, forming butyryl ACP. During fatty acid synthesis, the activation and elongation steps are repeated until the desired fatty acid chain length is achieved.
In conclusion, lipid metabolism, including beta oxidation and synthesis of fatty acids, is a vital process in living organisms by producing and storing energy to maintain homeostasis.